Alrighty, welcome everyone. We're gonna go ahead and get started today. Today we're gonna continue and build upon, hopefully wrap up some of our discussions from the previous two live streams where, we've been talking about uh, the process for doing machine learning, the basics for doing machine learning. We talked about the differences between supervised and unsupervised learning. We've talked about the differences between classification and regression algorithms. We've talked about the overall framework for approaching machine learning last time with the crisp DM methodology, and this whole time we've been diving into decision trees, using decision trees as a basic example of an introductory classification algorithm. And I've been promising we would get into the weeds under the hood of decision trees, but we've kind of kept on postponing that because our other discussions have gone long. Well, today's the day where we're really going to get under the hood, really understand exactly how decision trees make their decisions and exactly how they classify our data. We're not going to be burdened as much with data cleaning, things like we talked about last time. We talked about how at a bare minimum, every single machine learning algorithm needs to have no missing values and number two, have all the values be numeric before we can pass those values as inputs to a machine learning model, to an algorithm, right? And then it gives us those predictions back out as outputs. We're gonna jump through all that today. I've really simplified the code here. In previous notebooks, in previous discussions, right, we had we would have some code to diagnose the problem, then we'd have some code to fix the problem, then we'd have some code to confirm that we really had fixed the problem, right? So a lot of these things took 3, 4, 5 code cells. Well, today I've just put them really concisely in our notebook. But let me share this notebook with you so that you can follow along if you'd like. I'm going to make I'm going to make a copy of this. So this starter notebook will remain blank so that you can follow along with me if you like to. So you all as well can make a copy of the starter notebook and code along with me. We're going to be writing a lot of code today, so that might be a good thing to do. Okay, and then, so this starter version is going to remain blank. I'm going to make a copy of it in Drive, and eventually this new one that comes up is the version that we are going to be doing together live, something like that. I still do not have a great naming convention for differentiating between the starter notebook and the live notebook, and I have a notes version where I've already written out some things to refer to. So this live version, if you refresh the page as I'm going along, you should be able to see the code that I have written. There's that in the chat. Cool. Okay. So last time we were using this penguins data set. I told you how much I love penguins, and this data set in particular. I think it's such a good practice data set because it has a variety of different kinds of variables. It's got continuous variables, but it also has some categorical or discrete variables, right? And that was really our big discussion last time, particularly that our y variable column, in this case the species, because it has 3 categories. It's a discrete or categorical variable. That means we're going to be using classification algorithms to predict this species column. Okay, but let's get rid of some of the nan values. We're going to fill the sex column with male, and then we're going to drop any rows that have nans after we've done that. So we've gone from 344 rows to 342. Okay, and we'll do some of the quote-unquote categorical encoding, meaning taking these string columns and turning them into numbers, into numeric columns. We'll do that a little bit later on. I want to leave the nice written out names for now because I want to make some graphs with you. A couple of the graphs we'll have made last time, but we're going to have some new ones as well. And if I leave these written out string version of the names in here, when I build the graphs will automatically be labeled nicely. So after I build the graphs, then we're going to do the categorical encoding down here, and we'll talk about that when we get there. All right. Any questions before we dive in? So we're going to import Seaborn here. Seaborn is really built upon another library called Matplotlib. Matplotlib is one of the basic fundamental graphing libraries that exists in Python, and Seaborn provides us functions that make it so we can generate Matplotlib graphs with very little code. And one of these functions, let me make my font here a little bit bigger, is the histplot function. And we have to tell it our data frame, give it our data, and then we tell it what we want to exist on the x-axis, because we're going to make a histogram, hence histplot. Let's put the flipper length. Okay. And if I just run this as is, a couple of things happen. This is the flipper length for all penguins across my entire data set. I also get this funky output here at the top. I can suppress that output if I put a semicolon at the end of this line. And then I just get the graph. That kind of cleans it up a little bit. I can have this be made up of more columns. If I change the bins parameter to 20, I'm going to leave that at 20 for all the graphs we make today, but you can fiddle with it. Okay. We get a little bit more granularity if we do that. And then we can also use the hue parameter if we want this histogram to be broken up by some categorical column. In this case, we want to look at the species column because that's our Y variable that we are interested in. All right, and now we can see that really that histogram we saw before was made up of a few different distributions put together. We had a distribution for the flipper lengths of Adelie penguins, of Gentoo, of Chinstrap, all put together into that one larger graph. And this histogram is showing those distributions overlapping a little bit. So you have to imagine that these distributions are a little bit transparent. So we see a green Chinstrap distribution, a blue Adelie distribution, and a yellow Gentoo distribution. Okay. And I'm going to do this, I'm going to make the same graph with all four of the major continuous variables that we have: flipper length, bill depth, bill length, and body mass. And this is probably going to, auto-populate this for me. This is one of the nice new things in Google Colab and the advent of AI, is that it's pretty good at guessing and matching your code previously above, right? Because I put bin size of 20 and added the hue here, it then suggested that in later graphs. Okay, so we should end up with 4 graphs here. Bill depth being the last one. Okay, and you'll see that we talked about how the decision tree is going to try and isolate a portion of our penguins based on picking a cutoff, right? It's going to look at this distribution and say, if I separate all these penguins into 2 groups, right? Because I'm only going to split it one way or the other. Pick a threshold in the middle, say a bill depth of 17. Will that help me isolate one of these categories of penguins in particular? If I were to use bill depth, I could isolate the Gentoo penguins pretty well. If I were to use the bill length, I could isolate maybe the Adelie penguins pretty well. Body mass, I could also identify the Gentoo penguins. Same with flipper length. You see that the Gentoo penguins are a little bit of an outlier here, that, or a little bit more different from the other two. That's because Gentoo penguins are by and large a little bit larger than the other penguins. They have longer flippers, they have a bigger body mass, right? Bill lengths on the longer side, but Chinstrap penguins also have long bills, and they have kind of not as deep of bills. The bill depth here, I believe, is more like bill height. Don't quote me on that. Yeah, they're a little bit different. The Adelie and Chinstrap are pretty similar size-wise. Gentoo's a little bit off. Okay, now I want to show you something cool. Anytime we want to look at these graphs, I've got to scroll around to look at them. But because I have exactly 4 of these graphs, I think it would be really cool if we could use something called Matplotlib subplots to put these in a little 2x2 grid so we can see them all on the screen at the same time. Okay, and once you know how to do this, it's really not too bad. It can be a little bit tricky. First thing is we need to import Matplotlib, which I told you was the underlying graphing library that Seaborn is using, right? So we can use any Matplotlib code that we want to modify Seaborn code, and they will play well together. They will not interfere with one another, okay? So I'm going to import matplotlib.pyplot as PLT. And then I have this funny line here. Fig, x equals plt.subplots, okay? And I want a 2x2 grid. And then I'm going to give it a fig size. This fig size is just going to tell it how large I want the overall graph to be. And let's make it a 15 by 10. I may adjust that in a minute. Okay. So what this has now done is it's going to make a graph with 4 holes in it. Oh, I need another comma in here. Okay. I have a place for 4 graphs. Right. The figure is the whole background that these 4 graphs sit inside of, and then each of these 4 graphs is a different axis, or they're different axes objects, right? So I have 4 of them. So I can now take my his plot, right, and I can pick an axis or axes to put this into, right? If I pick 0, 0 here, then it's going to take my flipper length graph and put it here at the top. So this is row 0, row 1, and this is column 0, column 1. So the different positions, they're 0 indexed, right? So if I do that, now that should fill in this first graph here, which is pretty cool. And if I keep on going here, Google Colab is pretty smart, and it's going to put the other 3 graphs at 0, 1, 1, 0, and 1, 1. And now I have all 4 of the graphs on the same screen and visible, which I think is pretty cool and pretty nice. All right, so the other thing I was going to show you is that we have this funny output here. We can again suppress that with a semicolon, or we can say plt.show here at the end, which is actually the method I recommend for doing this. And then that funny little output at the top will go away, and we'll have just the graphs in a code cell all on their own inside of matplotlib subplots, which is kind of cool. So a very, very short subplots introduction there. Okay, but we've already talked about these graphs. I want to make a couple of new graphs with you today, right? Because this helps us look at the continuous data, right? And I can also look at the continuous data by doing something like df.describe. This will give me the summary statistics of these things, so I can now see the the max and the min value for each of these. Oh, that's a pretty big penguin at 6,300 grams, right? The smallest penguin is 2,700 grams, so 2.7 kilos, 6.3 kilos. The 50th percentile here is also known as the median, right? You can see some interesting things here. Just giving you a little bit more specific data about these distributions, but not exactly in a graph. Okay. Well, what if I wanted to do something with the island or the sex column, right? What if I wanted to compare the island of the penguins to the species of the penguins? Well, I can use a cool pandas tool for this called a crosstab. Crosstabs are good when we are comparing categorical variables to other categorical variables, right? These color-coded histograms are when I compared a continuous variable to a categorical variable, right? Because species is categorical, but these other 4 measures are continuous. Well, if I have 2 categorical variables, like, say, species and island, a crosstab is going to count these up and put them in a little grid for me, right? And the intersection of these grids says how many categories there are of how many observations there are that match both of those categories. So there are 44 Adelie penguins found on Biscoe Island. There were 55 Adelie penguins found on Dream Island. There were 68 chinstrap penguins found on Dream Island. And if you stare at this long enough, you'll realize that Torgerson Island only has 1 kind of penguin on it. Only has Adelie penguins. So when we're looking to make classifications, if I find a penguin and I know that it was found on Torgerson Island, probably help me make a good classification there. I'll be able to say, oh, it's Adelie, and I'll be right, according to this data set, 100% of the time, right? But one of the really cool things is that with pandas, I can make really easy and effective, really cool plots from these crosstabs very easy, right? So I make the crosstab, but then at the end of this, I'm going to say dot plot, okay? And by default, when you do dot plot, you get a line graph. And I found that line graphs are usually not what I want when I do this. But there's another parameter I can pass in here called kind, the kind parameter, and I can turn this into a bar chart, for example. Okay, so now this is showing us species on the x-axis, and then the number of, oh, and the island is color coded here. I think this is actually the opposite of what I want. I think I want to see island on the x-axis and have the species be the colors. And I think I can change that if I just switch the order of these two arguments right here. Okay, so now I can see the quantity of penguins by island. And I can see Adelie penguins on all three islands. Chinstrap penguins were only found on Dream Island, and Gentoo penguins were only found on Biscoe Island, right? And this graph tells us that the island says a lot about penguin species, right? The island is going to be a really helpful tool in classifying these penguins. Cool. So that's crosstabs, and we call this pandas plotting when we call dot plot at the end of something. Pandas is also using matplotlib to generate these graphs, but like Seaborn, it's giving us some simplified syntax so that we didn't have to draw all the rectangles and add the legend and do all that stuff manually. It's kind of inferring a lot of what we wanted to show up in this graph based on the table that we gave it, based on the crosstab we provided. All right. So I have a question for you. Why don't you think, as I type this out, why don't you think we've talked about the column, the sex of the penguin, very much so far, right? If I make this island, crosstab here, what does this crosstab tell me about the relationship between the sex of the penguin and the island it was found on? Can you make any vague or observations about it? And while you're thinking about that and maybe typing a response, I'm going to do the same thing. I'm going to turn this into a bar graph so we can look at it visually as well. Sometimes our brains are better at picking up patterns through graphs than they are through staring at tables. Okay, Caesar says about 50% male, 50% female for an island. Yeah. Across all the different islands, it's about 50-50, right? Now, in our data set at large, we have slightly more males than females, and we have even some extra males, because remember when we did our imputation of the missing values, of the NaN values, we said, oh, make those all male, right? So we have slightly more males than females, but part of that is an artifact of how we've cleaned up the data, right? What this tells me is that one of two things happened. Either one, the researchers tried to get an even 50-50 split of males and females. I doubt that's what they did. I bet they tried to have a true random sample in the penguins they selected. I bet it is hard to tell the difference between a male and a female penguin based on how they look. So they just grabbed a penguin and they checked what gender it was, what its biological sex was, excuse me. And I don't know how you sex a penguin, and I don't really want to know either, but like it must end up about 50-50. Like if you grabbed a random penguin, 50-50 odds, you grabbed a male versus a female. So I want to do something else. I want to explore, like, is there a strong relationship between the sex of the penguin and the species of the penguin, right? Did they tend to grab a lot more male Gen 2 penguins or something like that, right? So let's look at species. And the answer to that is also no, right? So I don't think the sex column is going to be a very strong predictor of species, right? And that's because the sex of the penguins is more or less randomly distributed with respect to our target, with our Y variable, the species column, right? And so while we're not going to throw it out, we'll still include this variable in, we'll still give this variable to our machine learning algorithm. I don't think it's going to be able to utilize it very well to differentiate between the species because the proportions are so even across all of these different species. And we're going to talk a lot about proportions today. The main statistical measure that a decision tree uses to tell how well the decision tree is classifying has a lot to do with proportions. So this is I'm trying to kind of bait the hook for a little bit later when we get into decision trees with this as well. Okay. Let's see. Okay, I won't make a couple more kinds of graphs here quickly. I don't want to take forever on this because I don't want to run out of time to talk about decision trees. Okay. I want to maybe make a scatter plot. Okay, scatter plot just puts dots in a grid here. And yeah, let's use some of the continuous variables. Let's use, like it's suggesting here, flipper length and body mass. I'm going to put I always like to put the data as the first argument here. And let's leave the species off for a second. Need a comma. Okay. So there's our scatter plot. Looks like body mass and flipper length tend to be correlated, right? The longer the flippers, the bigger the penguin. That makes sense. Okay. Now let's put that hue parameter back in and color code this by species. And now what do we see? We see our bigger Gen 2 penguins over here on the right and our similarly sized Adelie and chinstrap penguins on the left. Pretty cool. However, what if I change this and instead of having the hue be species, I had the hue be sex? Now blue is male and yellow is female, so we can see within the Gen 2 penguins, males are bigger than females. And within the Adelie and chinstraps, we can also see that males are bigger than females. Right? If we look at those two clusters on their own. Right? In order to make this even more clear, let's plot only one of the species of penguins on one graph at a time. Right? So I need to first select maybe only the Adelie penguins. So I can, let's see. Oh, it wants me to do all of them. That's fine. So this says, if the species is Adelie, grab only those rows from the data frame and save it to this variable. We'll do that same thing with chinstrap and Gen 2. Okay. And now I just change my data frame here. Instead of plotting all of the data, I just plot the Adelie data. Okay, we see that males are bigger than females when it comes to Adelies. So maybe a researcher could have tried to randomize the size of penguins they were grabbing a little bit. We'll do the same thing with chinstrap. Not as many chinstrap penguins. And Gen 2. And this is another reason why we're not getting rid of the sex column. Because while the sex column is not very correlated with species, it is highly correlated with some of the other X variables. And so it could have some benefits in that way, right? If we only knew the sex of the penguin, well, that would tell us something about the penguin's size, which would then tell us something about the penguin's species, right? So kind of vicariously gives us some information. But you can see the bigger the penguin, excuse me, that male penguins are typically bigger than female penguins. Okay, last example here. We're going to make a scatter plot again. But instead of looking at this across two different continuous variables like body mass and flipper length, what if we only looked at it across one of these, right? So our data is still going to be Adelie, just as an example. And we'll have our X variable be, I don't know, body mass. We could really pick any one we want here. Well, we need another Y variable. What if I set that Y variable to be a bunch of zeros? Okay, so I'm going to say, hey, give me a list of zeros, and then we're going to multiply that by however many Adelie penguins we have. So there's going to be a list of a whole bunch of zeros, as many zeros as we have Adelie penguins. And what that's basically going to do is it's going to plot these on the x-axis, on a number line. So there's all the Adelie penguins, but we're only looking at the distribution of body mass. Again, we can throw in the hue parameter according to sex. And now we can really see the male-female difference. Basically, we've taken all these points and we've squished them onto the x-axis. Okay. And something that I like to do when we put them all on a line like this and they're kind of overlapping, I like to make them a little bit transparent with the alpha parameter. So we'll set alpha to 0.33. And now the darker points are in places where I have more data points, and the lighter ones are where I may have just one observation. And where it's kind of brownish, that means I have blue and yellow points on the same spot, kind of overlapping. And you can see maybe a little bit of a round numbers bias in this distribution as well. So that can be a cool way to like really make it clear, hey, males are bigger than females, and it makes it maybe even more stark than on a two-dimensional scatter plot. So now we know even more about these penguins. Let's see which of these things that we can see with our eyeballs, right? Our brains are these amazing pattern detection machines, and that's why making graphs is useful, right? Because I can make this graph and my brain immediately goes, oh, those bars are all about the same size. I can make this graph and my brain immediately goes, oh, there's more blue dots over here than there are yellow dots over here, right? That's why making graphs is useful, because our brains are good at detecting patterns. And so once we understand the language of graphs and data visualizations, we can notice things at a glance. And that's the goal, right? Is to be able to communicate things very quickly about really quite a lot of data points, a lot of information, but communicate one major insight in a snapshot. Okay. So that's kind of a quick rundown of some what I hope are useful and interesting data visualization tips for you using a mixture of Seaborn, pandas plotting, crosstabs, matplotlib subplots, some really important and really useful tools to have in your graphing tool bag. Okay. Any questions before we move on and start digging into decision trees more? What you maybe came here for last week? Finally get around to it. Okay. So now I'm going to categorically encode the island and the sect. We're going to turn those into numbers. They're just going to be integers to represent the different categories, right? And we're going to do species a little bit later. We have to do species before we make some graphs later on, but we don't necessarily have to do it before we pass the data to our machine learning algorithm. Okay? Our y variable does not have to be categorically encoded. It can still be a string value, but everything else has to be numbers before we pass it to our machine learning model. Okay? And then on Tuesday of last week, I did a train test split in this fashion with you. I don't know if you remember that, can think back to it, but we divided the data set into train and test, and then we split the data set into X and Y. Okay? We're going to do the opposite of that today. It'll have the same result, but this is probably the more traditional approach, where I split the data set into my X matrix, so-called, and my Y vector. That's why this uses a capital X, because in linear algebra, a matrix is represented with a capital variable, whereas a single row, single array of data, a vector of data, is represented with a lowercase variable. And then we're going to use this tool from scikit-learn, called this function called train test split, to split it into X train, X test, Y train, and Y test. And if you are like me and you have a hard time remembering the order of these variables here, remember that X comes before Y, and you always have to train a model before you can test it, before you can do that validation. Okay? And that might help you remember the order of these here. X before Y, train before test. I'm going to take 20% of the data points here and put those in the test data set. Everything else is going to be in the training data. And if you use the same random state as me, you should get exactly the same results as me as we move along. And we talked about how having a baseline is good. But we built our baseline using the whole data set. We really should have done it only using the training data. Okay? So my baseline would be whatever the majority class here is. If I predicted that for everything, how often would I be right? And we did kind of a funky calculation here. I'm going to show you, in fact, a simpler way to do this. When I'm doing value counts, it counts up the number of each species. But there's a parameter here I can pass where I can say normalize equals true, and it will turn these from raw counts into percentages. Okay? So I can see that now Adelie is 42%, and I can just get whatever the largest of those percentages is, and that's my baseline. Okay. Oops. This is the largest percentage of, well, the percentage of observations that make up the largest Y variable category. In other words, the class. Okay. So there's our baseline. We want to beat 42%. I think the other day we had 44% for our baseline. This is a little bit different because we have a slightly different random subset of the data set today, right? If I change this to 43, I'm going to get a different random 80% of the data set in my training data, and I'm going to get a slightly different percentage here. Every time I change this integer, I'm going to get a different 80%. So now it's 44%, 45%, right? So it's not an exact baseline, but it still is a useful baseline. Okay. All right. That's enough about baselines. We're gonna skip our dumb model as well. We know what our dumb model is gonna do. Yeah, let's just skip that. We talked about that last time. All right, so now let's use scikit-learn to fit a decision tree. We import it. We make one version of that kind of model. We give it a random state so that its output will be consistent between my code and your code. We're going to fit it on our training data and then validate it on our test data. And it tells us our model is accurate about 95% of the time, which is a huge improvement over the baseline. And let's talk about how did the decision tree actually do this now. Okay. Well, there's one kind of decision tree visualization that I really like, but the tool to make it does not come pre-installed in Google Colab. If we run this pip install command with this bang symbol at the front, this exclamation point, this will reach into the computer that is fueling our Google Colab notebook, and it will install this tool on that computer for us. Okay. So it's installing all of it. And we don't need to keep any of this output here. So once it's done installing here, I'm going to clear that output just so it makes our notebook a little bit cleaner. And then we can go ahead and use it. But you notice if I try and generate this right now, it's going to get mad at me because it needs a list of the names of the features of our data set. And those are the column headers of our X-trained data set. Right? But it's not going to like it if it's here as what's called an index. So I have to cast this to a list, and then it will be happy about it. And then I'll save that to a variable features, which I'm already using here. Feature names comes from this features list. I also need same thing, but with the numbers of species. And I can hard code it here, but I could also grab that from y train.value counts. Okay, and I want the left-hand column, I want the names, so I want the index here. And again, it's not going to like it as an index, so I still have to say to list. And there we go. There's the species, as I've called it. So those are two lists we just need to help make this visualization down below. And as I run this, let's say it says features is not defined. Features should be defined. Make sure I've run that code cell. It says, I'm still not liking this because you gave me a string. Okay, so where my machine learning algorithm, my decision tree classifier, doesn't care if this species column is a string, my visualization does. My visualization doesn't want the y variable to be a string. Okay, so for the sake of the visualization, not for the sake of the machine learning algorithm, I'm going to go back And we are going to now categorically encode the species column. It shouldn't change anything with regards to our model's predictions or our baseline or any of that. It should still be 95% accurate. All that should be the same. But now the species is 0 to 1 rather than the written out names. And now my graph should work. Okay, and we saw this briefly on Friday. We saw that, hey, the decision tree saw what we saw in the graphs. It said, hey, if I make a cutoff here, then I should be able to isolate what I think is the Gen 2 penguins really well. So this is the problem, is that now species is 0, 2, 1. But I think, yeah, so 1 is chin strap. So I isolated the chin strap penguins really well. 0 is Adelie, chin strap is 1, Gen 2 is 2. Okay. And I want to talk with you about how did the machine learning model know to use flipper length to make its first split? How did it know to not use, scrolling all the way up, how did it know to not use bill depth or body mass? Those would also be good at isolating the Gen 2 penguins. Why did it pick flipper length, number one? And how did it know to pick exactly the cutoff of 207.5, right? Did it know that that was the best cutoff? How did it know? How did it calculate that? And that's really what I want to address with you with the time we have remaining. I'm going to generate a second decision tree visualization here. This is one that comes from scikit-learn itself. I don't think it's as pretty as this visualization up here, but it gives us a little bit more information. Specifically, let's see what I can do here. I want the text, I want the font to be a little bit bigger. That made it worse. Oh, and even worse. Still not great. This figure size is just allowing the graph to be bigger in certain directions. I hope you can read that. But it gives us a little bit more numeric information than this one does. This one tells us the cutoff really well, and I think it helps visualize why the decision tree made the cutoff there. But this one also tells us a really important piece of information called the Gini impurity. Okay, that's what I want to talk about with you today. Okay, before we get into that, we have to establish a little bit of terminology, right? First off, these are called decision trees, but the tree grows downwards. It's kind of weird. Instead of growing upwards, it grows downwards. Ask computer scientists why they decided to make trees like that. You can think about it as the roots of the tree if you want, but we have the root node here. That's our whole data set, right? And whenever our decision tree makes a split, say it picks a flipper length of 207. If your flipper length is less than or equal to 207, then those data points will be put on the left side. And if the flipper length is greater than 207.5, they'll go to the right side, right? So left is less than, right is greater than, right? Or if it were a categorical value, then it uses true and false. And if a categorical statement is true, then it'll go to the left. If it's false, it will go to the right. So an example of this is island, right? So it said, well, this is still not a great example because the island we made into numbers. So it's saying if your island is greater than 0.5, which basically means if you're on Dream or Bisco Island, I think, then you go over here. And if you're on Torgeser Island, you go over here. I might have those islands slightly off, right? But it's true to the left, false to the right, generally. Or greater than to the right, less than to the left. Okay? Of the given cutoff. But our whole data set, we call that the root node. As we go along, we have parent nodes and child nodes. That's just a parent node is closer to the root. And then once we get to the very bottom, these are called leaf nodes when they are perfectly categorized, right? So the goal of the decision tree is to make splits, to keep on splitting the data until we get totally pure nodes. That means nodes that correspond to only a single category, right? So this is all chin-strapped penguins, and this is all Adelie penguins, and this is all Gen 2 penguins, and it's trying to isolate them by picking the best places to make splits. Okay. And then later on, we can then classify those penguins by following the branches of this decision tree. Oh, it's a penguin that has a flipper length less than 207, then I'm over here. Has a bill length greater than 43, well, then I come over here. And it's on Dream Island, well, now I'm over here. And has a bill length less than, you know, and I can follow the decision tree down based on the different thresholds and the different cutoffs. Okay. Well, how does the decision tree do this? Well, through a certain metric. That metric is called Gini impurity. Gini impurity. All right. And here's this fancy pants mathematical calculation for it. It is a lot simpler than this math notation makes it look like. And that's really my goal today, is to make you think about Gini impurity and go, oh, that's not too bad. Okay. And then I'll give you a little bit of code that could calculate exactly which column should we have used in the first cutoff. Why did we use flipper length? And then where exactly should we make that cutoff within the decision tree? Okay. So when we talk about Gini impurity, we have 1 minus the sum of each of the squared proportions, P sub I. So what is sub I here? Sub I means each of the different categories, right? So if I were to expand this summation, it looks something more like this. 1 minus the percentage of observations that are Gen 2 penguins, the percentage that are Adelie, and the percentage that are chin strap. Okay? So right off the bat, our data set already has a level of purity to it. And that level of purity is the degree to which the data set is made up of one single category or is across many different categories, right? So if I take my Y train and do value counts, okay, and let's see, let's also print ytrain.shape. Let's do that above the value counts. Okay, so I have a total of 273 observations, and these are the different species. Okay, well then my Gini impurity is going to be 1 minus the proportion of all of my observations that are Adelie penguins, which is 116 divided by 273. In other words, the percentage of my total observations that correspond to Adelie, right? And I'm going to square that value. Okay? And I'm going to do the same thing. I'm going to subtract 100 divided by 273. That's the percentage of, what is 2 again? 2 is, Gen 2 penguins. 2 Gen 2. Okay, I can remember that. We're going to square that as well. And we do the same thing with the chin strap penguins, right? And this is the Gini impurity of my data set at the beginning. 0.64, and if you round this up, 0.642. I want you to see that this number is exactly the same number that we have here. The Gini of the whole data set, 0.642. Okay? Now, I want the level of impurity in my data set to go down, right? Because I want these final leaf nodes, I want them to be as pure as possible. So I want to increase purity, I want to decrease impurity, right? And so Gini is a measure of impurity. We want less impurity. We want the Gini impurity number to go down. We want to minimize it, okay? Have to wrap your head around that a little bit. Okay. I want to show you that we can get this same calculation in a slightly different way. Okay. I can subtract each of the squared proportions, each of the squared percentages from 1. That's one way I can do it. I can also subtract all of them added together. So I wrap this in parentheses and I'm adding them all together before I subtract them from 1. That will give me exactly the same number. So just a little mathematical notation trickiness there. These are equivalent calculations. So I can subtract the sum of the squared proportions from 1 is really what's happening here. Now, why am I squaring these proportions and why am I subtracting it from 1? Well, I'm subtracting it from 1 because in machine learning, our metric that tells us how good our predictions are, we typically want it to get lower. Lower is better. We call this a loss metric or an error metric, and we want less error, we want less loss. So if I just left it like this, well, this is Gini purity, and the Gini purity can go up to 1, but the Gini impurity goes down to 0. So really I'm just subtracting it from 1 so that it goes downwards. Down is better. Okay? And I want you to imagine a couple of things here. Oh, let's calculate the Gini impurity for the first node cutoff. Oh, we're not going to do that quite yet. What if I had exactly the same proportion of observations for all three? What if exactly 1/3 of my observations were of each species? Well, then what would be the Gini impurity? Well, 1/3 squared is 1/9. 1/3 squared is 1/9. 1/3 squared is 1/9. That would be added together 3/9. So this should be 1 minus 3/9, or 1 minus 1/3 should be 0.666666, right? So if my data set had exactly even classes, exactly 1/3 of all the penguins were of each species, well, then if I were to pick a penguin at random, the odds of me being wrong are 66%. Okay? I'm going to say that again. If the proportion of each species was equal, 1/3 each, the odds of me making a wrong prediction, if I were to randomly select a penguin species, are this. Okay. The odds of making a wrong prediction if I make a random selection. Okay. If I'm using my dumb baseline model, it's kind of like this. Well, my dumb baseline model wouldn't have exactly 1/3. It would have the baseline proportion. So 0.64, that is the odds of making a bad prediction if I have my dumb baseline model, right? That is just predicting a deli percentage of the time, and Gen 2 percentage of the time, and chinstrap percentage of the time. I want to contrast this with, what if I had a model that had a lot of what we call imbalance in the y variable, right? Say that like 99 out of 100 observations were of one class, and we had, well, this will have to be 0.5% were another class, and 0.5% were the other class. Well, then now my purity is really close to 0 because my data set is already pretty pure. My data set is already mostly one class or one species of penguin in this example, right? And so our Gini impurity can range anywhere from, well, 0.666 for if we're doing classification with 3 classes, this is as high as it can get, and the lowest it can get is 0, right? And it's going to be somewhere in between those two numbers. The lower, the better, because if I'm making a split that leads to pure leaf nodes, well, then that's a more useful split to be making. That's a split that's going to lead me to make correct predictions more often than some other split. Okay. So I hope you see that the calculation here really is not very crazy. You can type it out and you can do it by hand. Okay. But we can use some pandas and Python tricks so that we don't have to write this out every time. we can do a much faster calculation. It'll look a little bit nasty, but it's going to be a one-liner that will work for any cutoff. Okay. So I want you to imagine, let's calculate the Gini for this first flipper length cutoff, right, of 207.5. This is what the decision tree chose. And we're going to try and see why did the decision tree choose that specifically. Okay. And in order to do this, I'm going to actually combine back together my X-train and Y-train data sets. And I'm just going to call it train. This is going to be so that it'll be more easy for me to relate flipper length to species because they'll be on the same row of the same data set. So I can put these back together using this pd.concat function. And I give it a list of data frames I want it to squish back together. And this X-train and Y-train along the column axis. I want it to add the Y-train column back on to the X-train columns. Okay, and this is just for convenience here, but now I've got them back together. Species columns back on here. Okay, and now what does this split do? Well, the split divides my data set into two subsets. I'm going to call this the left portion of my data set and the right portion of my data set. Okay, left is less than or equal to that cutoff, right is greater than that cutoff. Okay, and what my data set's really doing is it's trying out a bunch of different cutoffs, a bunch of different ones. Okay, and it tries out pretty much every single cutoff that it is possible for it to try out across the whole data set, and it calculates the Gini impurity across the left and the right splits for every single cutoff. And to judge whether or not that cutoff is leading to purer nodes, it's going to take the average of the Gini impurity of the two child nodes. Okay, so we make the split, number one. We calculate the Gini impurities of left and right, and then we average them, and that tells us how good of a split this is. Basically, is what's going on. Okay, so let me show you some tricks to make that calculation not too crazy. Okay, so we're gonna pick our cutoff here. I'm gonna say that that's 207.5. Okay, and now we want a left data set, and let's see, whatever is less than or equal to the cutoff, that goes in left, and greater than the cutoff, that goes in right. Okay, notice we have 168 to the left and 105 to the right. That should match what it says here. Oh, it made it bigger. I guess that's kind of good. 168 samples on the left, 105 on the right. So that's good. We've matched the split here. We've done the split successfully, same as the decision tree's done it. Okay. But now I need to calculate the Gini impurity of left and right. Okay. Oh wait, so let's see. Recombine, I meant to do this down here. Split into nodes based on the cutoff. Just to follow the structure of my notebook. Okay, so there's the cutoff. Now I'll calculate what are called the leaf impurities, the two impurities separately. So left impurity, well, let me show you a trick. If I take left and then I look at the species column, and I do a value counts, that tells me the total number of each species. But there's another parameter I can pass in here. I can say normalize equals true. We saw this above, and now turns these into percentages. So these numbers are now the proportions that I want to put inside of here. First number, second number, third number. I want those to be these. First number, second number, third number. Okay. I can isolate just these numbers by saying dot values. Dot values is always the second column in an output like this. So there's the three numbers that I want to be in those values. Well, I want to square each of those values. Now they've each been squared. Now I want to sum up those squared values. And then I want to subtract that from 1. So this calculation is equivalent to me taking each of those proportions and putting them in these parentheses, squaring them, subtracting them from 1. It's a little bit nasty, but if you understand what a value counts does, that normalizes getting the percentages, aka the proportions, dot values is isolating that column, then we're squaring all of them, summing them up, and subtracting from 1. This is the left Gini impurity. Okay. The right Gini impurity would be the same thing, except instead of left here, I do right. Okay. So there's left and right. Okay. And if I want to score for this particular cutoff, what I do is I average those two. I average the impurities of the 2 leaf nodes to get a score for the quality of the split that I have made in the data set based on the selected cutoff. And remember, lower score is better. Lower score is better. Okay, so let's average these two. There's a really easy way. If I know I'm always going to have 2 numbers, then I can average these by adding them together and dividing by 2. And so this is the Gini impurity for my cutoff. .2745. So 0.275. Let's see if we see that number. Okay, so here's the Gini. Oh, there's the Gini for the left, and there's the Gini for the right. It doesn't actually show us the average on here, but we calculated that 0.44 and that 0.109. Okay. So we now have all the information here, and the value here, that list of values, is the same thing as our value counts dot values. Right? So we have all this information. We've calculated it quite easily. Okay. So what my decision tree is telling me, it's telling me that this cutoff on the flipper length column of 207.5 gave us the lowest Gini impurity of any other split we could make anywhere in the data set. Any split that we could have made in the body mass, any split that we could have made in the bill depth, this is the best of all of those. And the real question is, how did the model know it was the best cutoff? Well, it tried every single possible cutoff, not just within flipper length, but within the entire data set. And it calculated the average Gini impurity of the 2 leaf nodes that were generated from those cutoffs. And it stored that somewhere, and then it found whatever the lowest one was. And that's where it made its very first split in the decision tree. Okay. So we're about at time here, but I want to, well, we are at time. If you're willing to hang on and keep on watching. If you need to jump off at the hour mark, jump off. But I'm going to finish coding with you a very rough implementation of how might the model have decided upon A, flipper length, but B, 207.5. Basically, we're going to calculate the average Gini impurity of the 2 leaf nodes of every single cutoff we could possibly make given our data set. Okay? Let's see. So what if we wanted to do that ourselves? Test every single possible cutoff? That's what we're going to do right now. Okay. So let's start off. Let's just do it for flipper length first, and then we'll make it happen for every column at first, right? So what we need to do is we need to look at the flipper length values. Okay. And if I sort these, it's going to automatically, let's see, has no attribute sort. Well, let's do this with NumPy. And we'll say np.sort. Okay. Oh, you know why it's not working? That's one way to do it. It's not working because the pandas function is not sort. It is sort values. Yeah, so this is the same thing as this. I like the NumPy version because it shows us all of the numbers. Okay. And you'll notice that we have a lot of repeated numbers here. We don't care about repeated numbers. We just want the unique values, right? So I don't need to include 200 in here 4 times. I just want 200 in here 1 time. So not only I'm going to sort this, I'm going to grab just the unique values and sort those. It's a much smaller list. Okay. Now what does my model do? It needs a cutoff. So it wants to use cutoff as the cutoff values numbers that are not in this list. In fact, it wants to use all the numbers between these numbers, right? And so I have 172 and 174, so one of my cutoffs could be 173, exactly in between those two numbers. 175 would be exactly in between these two numbers, right? So I need to look at every single pair of numbers in this and find the number that goes in between those. In other words, find the average of those two numbers. And that's a little bit tricky, but doable, okay? So we're going to do that with a for loop. Average of each pair of numbers to generate possible cutoffs. Okay, and I'm going to use a little for loop here. The more advanced you get with your code writing, the less like raw for loops you should write. This is definitely not the best or most efficient implementation, but I hope it's an implementation that is somewhat easy to follow if you're interested in the raw implementation here. So here's the thing. Actually, before I write that, this sorted list, I accidentally deleted a parenthesis, has 53 numbers in it. But the number of cutoffs that I want is 1 less than that, because the cutoffs are always going to come between these. So actually I'm going to have 52 cutoffs generated from 53 unique values. So I don't want to just loop over all of these values. I want to loop over however many of these values I have. Let's call this sorted values. And then we'll look at the length of sorted values. All right, that's a little bit cleaner. That needs to be an equal sign. Okay. I want the length of sorted values -1, because the length is 53. I only want to calculate 52 cutoffs. Okay. And then once I calculate those cutoffs, let's just, for a minute, let's print out the I. All right, so the I goes from 0 to 0 to 51. Range, length, sorted lengths. Oh, right, because it starts at 0. Right, so because it starts at 0 and it ends at 51, there are 52 numbers here, but it ends at 51. So 0 to 51 makes up 52 numbers. Okay, and the same thing I can do is I can say sorted lengths I and sorted lengths I plus 1. This should be every pair of values. Not sorted lengths. This is called sorted values. It's called sorted lengths in my notes. That's why I've got it on the brain. Okay, so here's all the pairs of numbers that we want to calculate. But we don't want the pairs. We want the average of each of these two numbers. All right, so my cutoffs are going to be stored in this blank list. And I want to add these two numbers together, right? The average of two numbers is always adding them together and then dividing them by 2. I can only do that when I know I'm going to have exactly 2 numbers. Okay. These are all the cutoffs. And you'll notice within this list is 207.5. Okay. So these are all the different cutoffs I need to try out. I need to calculate, split into leaf nodes, calculate their separate Gini impurities, and then calculate their average Gini impurity for every single cutoff here. And these cutoffs are only for the flipper lengths. Only for the flipper lengths, okay? Instead of printing this here, let's say cutoffs.append, and we'll throw those into this list. Okay, so now I have a list of all the cutoffs that I want to try. I'm going to print it out because when I print it out, it shows sideways and doesn't take up as much space in my notebook rather than printing out vertically. Okay, so I've now calculated all of my cutoffs, right? Well, let's see here. Now from those cutoffs, right? So for cutoff in cutoffs, for each of those cutoffs, I want to generate a left and a right. Just like I did above. And then I want to get the Gini impurities for left and right, which is here. So by building up by step, we've already done a lot of the work above. And then I want to get average Gini impurity. Which is left plus right divided by 2. Okay. And this will give me the cutoff for that, the average Gini impurity for that given cutoff. What should I call this? I'm going to make a list here to store all these. And I'm going to append all these to a list, but I'm going to append these as what's called a tuple. Basically, I'm just going to put them in parentheses. So I'm going to store both the cutoff and the average Gini impurity. Okay. So here's all the cutoffs on the left, and here's all the different averaged Gini impurities on the right. And if I can see it starts off pretty good, then it goes up to about 0.46. Remember, we want the lowest number. There's 0.274, and that is the lowest number anywhere in this list. Right? So that is why 207.5 was chosen as the cutoff. Okay, kind of crazy that every time a decision tree makes a split, it is looking across the entire data set, finding every single place it can make a cutoff in every single variable, testing, actually making the cutoffs in all of those, calculating the resultant Gini impurities of the 2 leaf nodes, averaging the Gini impurities of the 2 leaf nodes, and then trying to find the minimum, the lowest one. That's how a decision tree decides where to make its splits. Okay. Well, we want to do this again. Okay. So that's how it knew it was 207.5. I guess if I really want to, I can put this into a data frame. Things are just a little bit easier to work with when they're in data frames. I had a syntax error there. Okay. And then I can easily find the minimum. If I look at the Gini impurity column and get the minimum of that. What if I don't want just the minimum value? Because I mean, that's great. I want to know what the cutoff is. So if I say that this is called the index minimum, this is going to tell me whichever row has the smallest Gini impurity. Best cutoff index is that. And then I can I have to do this again. Okay. So here is the row that corresponds to the best cutoff. That's a very long-winded way of selecting just this. But programmatically saying, oh, which one of these is the smallest? Oh, if that one's the smallest, print it out. So I'm printing off getting the smallest. The best cutoff. Okay. Well, how did the model know that flipper length should have been first? Why didn't it start with body mass or something else, right? Well, it did it across all of the columns. Okay. So here's where this gets even crazier. I'm going to for column in xtrain.columns. Here's all of my different columns. I need to test the cutoffs for all columns now at the same time and get the lowest across all of them. Okay, which basically means this. I'm doing the same thing as above. I'm doing this, but I'm doing it for each individual column now. Right. So instead of doing it here for flipper length millimeters, I'm now just doing it for whatever column I happen to be looping over. Oof, yuck. What if we do this? Genie cutoffs. What if we add the column to this? There we go. So you notice here's all the different island cutoffs. Here's all the different bill lengths. Here's all the different bill depths. And this seems to be a little bit wrong because that's the same cutoff for each individual one. So I don't think this is quite right. Let's back up here. Let's think through this. So I'm looking at each individual column. And what do I want to do for that column? I want to get the potential cutoffs for that column. And I'm going to do that I want this to be the sorted values. Okay, so I need to sort each column first. So sorted lengths. No, I should just call this sorted values because it's not always length. Sometimes it's weight. There we go. So I'm getting the unique values, then I'm sorting them. So this is the sorted values for any given column. Okay. And then I can use that to generate the cutoffs. So this should now be sorted values minus 1. And then it's seeing my code above, and so it's filling this in for me. It's one of the great things about the AI era, right? Now I'm getting those potential cutoffs for me, being the average of each pair of numbers. Then for each cutoff in the potential cutoffs, I want to make the cutoff, which means getting a left and right. Get leaf node Gini impurities, which is left Gini impurity and right Gini impurity. And then I want to get the average leaf node Gini impurity. This is a lot of code, but believe it or not, this is a very simplified implementation of this. And that's why it's not very efficient, but okay. So I finally got that. I should now have the average Gini impurity for every cutoff of every column. Let's see what happens if I print those out. Okay, so for the island column, there's only two options, 0.5 and 1.5. Well, that makes sense because there's 3 islands, so I should have one less than that number of cutoffs. So I have 2 cutoffs for 3 islands. If I scroll down and find the sex column, which I think is last, I should only have one cutoff, right? 2 categories should lead to 1 cutoff. Okay. And on the right, I have the Gini impurities. So what my decision tree is telling me is that the smallest Gini impurity out of all of these was flipper length of 207.5. I should not find any Gini impurity that is less than this one. That should be the minimum of all of them. So let's throw all of these in a data frame. We'll do the same thing, find the minimum across all of these. Let's see, instead of printing them here, let's say, we'll make it even all the way outside of here. So all cutoff Gini impurities. That's a blank list. And then we will append these 3 as tuples. And the reason why I'm putting them in tuples is once I have a list of tuples, it's really easy to make it into a data frame. Okay? Tuple meaning these things are held in parentheses. So now all cutoff Gini impurities I can throw into a data frame. And so this is all Ginis. Okay. And if I want all Ginis dot, well, I want the Gini impurity column. And I want the index minimum. This will tell me the position of the row that is the smallest. All Ginis, Gini impurity index minimum, key error, Gini impurity. Saying it doesn't see a column named this. So I misspelled impurity. So it says row 256 is the best cutoff. In the whole data set. Okay. So all Ginis dot lock that's the best cutoff row index. We'll grab that here. And what do you know? It's in the flipper length column and it's 207.5. Whew. So that's what our decision tree is doing. It's looking at every possible place it can make a cutoff, testing them all out, picking the best one, and that's where it makes its first cutoff. Well, then what does it do? With the left-hand data set, it does it all over again. It looks at every single cutoff it could possibly make here, and then it picks the best one. And with the right-hand data set, it looks at all the possible cutoffs it can make with this remaining portion of the data set, and it makes the best one it can find over here, right? And you'll see as we go down, let's see, can I make this smaller? As we go down the tree, the Gini impurity values are always getting smaller because the model nodes are getting more and more pure. And whenever I get to a leaf node, that leaf node always has a Gini impurity of 0 because that node is perfectly pure. That's what makes it a leaf node. There's no more splits to be made because all of the samples in that portion of the data set are all of the same Y variable category. They're all the same species in our example. Okay. Now, decision trees aren't perfect. They will sometimes make wonky splits because they're not the smartest tool. Decision trees are what we call a greedy algorithm, meaning that they're going to make the very best split they can find first, not taking into account what happens afterwards, right? So the first split is the best one that can be possibly found. Second split is the second best one, so on and so on. The decision tree can't think ahead multiple steps and say, oh, if I were to split here, I'll be able to make a better split someplace else. It doesn't think about what happens in the later levels. It just does the best thing it can up front, and then the later levels face the consequences of that. So this may not be the very best decision tree that it is possible to create, but the number of values that would have to be tested if the decision tree had to think ahead a step or two would combinatorially explode and be intractable. But there are a few things we can use to kind of rein in our decision tree and to keep it from going too crazy, right? You'll notice that our decision tree here, one example of it when it may be making some not perfect classifications is after it splits here based on bill length, it almost has a pure node. Okay. Splitting here on bill length, let's see if I can show you better here. Okay. It has two penguins of one species, 49 of one species, and one penguin of the other. So it almost has a pure node. It just still needs to get rid of these two random penguins and this one other penguin, right? And so it makes some kind of weird splits just to get rid of the one penguin first and then to get rid of the other two. And look how many splits it has to do to get rid of those extra 3 penguins. Split off the one, split off one more. This split actually doesn't really even lead to any increase in purity. Actually, maybe it does. Oh, so we were able to isolate 48 of the 49 penguins here. We still have 2 penguins left, right? So this last split is kind of silly, right? Like, what are the odds that a penguin has a small bill, is on one specific island, but is also not on another island, and then has a short bill and a small body mass? Like, it's getting very overly specific. We might say that our model is overfitting. It's fitting to noise in the data rather than actual signal, because all that it knows how to do is make pure nodes. That's all that it knows how to do. And so it's going to do that at all costs, even if it has to make some pretty wonky decisions along the way, some overly specific decisions. Okay. We can rein in our decision tree and do what's called pruning by saying, hey, decision tree, don't make a leaf node unless that leaf node has more than X number of observations in it. Right now, it's making a lot of leaf nodes that have just one observation in it. This leaf node has 4, this one has 3, this one has 1, this one has 8, this one has 1, this one has 1, this one has 1, this one has 2, this one has 4, right? So I can actually say to my decision tree, and if I look at this in scikit-learn decision tree classifier, there's some extra parameters I can pass. Min samples split, the minimum number of samples required to split in an internal node. Min samples leaf, the minimum number of samples required to be at a leaf node, right? Or also max depth, the max depth of the tree. If none, then nodes are expanded until all leaves are pure, or until all leaves contain less than min samples split samples. So there's some parameters here I can use to rein in the overly specific decisions that my decision tree is using. So let me just throw a couple of those in here as an example. It's still going to do all the same operations. I'm just constraining it a little bit. Oh, that's the starter notebook. Okay. So here, when I make my decision tree classifier, I'm going to say max depth, set that to 3. And then let's also do min samples split. And we can test out different values here. Let's set it to 2. So there has to be at least 2 observations in order to make a leaf node. Okay. And this is going to make it so our tree is much smaller and is not making overly specific decisions. It's not going to be quite as overfit. We have a slight accuracy decrease on the training data, but we should overall hopefully generalize a little bit better to the training data. Okay, let's look at this again. Okay, so what we've just done is we've said we've just pruned the decision tree. We still have the same original cutoff, but we haven't allowed the the leaf nodes to be as wonky and as crazy. You'll notice that the tree is much simpler, much more straightforward, and less overly specific. And this may be a decision tree that generalizes better to unseen data than the one we looked at first. If we had more time, we would do something called hyperparameter tuning, where we would try out a bunch of different values here and see what gives us the absolute best validation accuracy. What set of parameters here that control the structure of the tree gives us the absolute best validation accuracy. We're not going to take the time to do that today. We can go more into hyperparameter tuning at a later time, as we're already over time here. But I just wanted to point out that we don't have to live with the default decision tree that gets generated here. We do have some control over the structure of the tree, bypassing some of those extra parameters when we fit the model. It's still using Gini impurity in exactly the same ways. We're just saying, hey, you can't make a leaf node there. That leaf node's too small. Or we're saying, hey, you can't make more than 3 levels of splits. That's really all that we're saying to our decision tree here. And that's called, again, pruning. Haha, tree, pruning a tree, right? We're cutting off some of its branches, so easy to remember. Okay, that's as far as we're going to go today. We could also write a function that will continue to recursively split and find every little branch and every little leaf node here, but I don't know if that's really the best use of our time here. I hope you can see that Gini impurity is not a very complex calculation. You square this, it's the sum of the squared proportions, and then subtract that from 1. 1 minus the sum of the squared percentages of each class. You do that for each leaf node, average across the pair of leaf nodes, and you check every single possible cutoff in the whole data set in order to determine where you're going to split at. And that's how this greedy little decision tree tries to classify our data, and it does a pretty darn good job. Okay? And the reason why decision trees are important is because they are used as building blocks for other models. Things like random forests and other ensembles of trees are some of the most powerful supervised learning algorithms that we have. And so understanding this underlying building block is going to give us a real leg up and a real head start when it comes to understanding some of those other more complex and honestly slightly better algorithms like decision trees or other kinds of boosted ensemble models, which that phrase might not mean anything to you right now, but things we can talk about in the future. All right, if you hung around for the end of that, thanks for sticking around. I want to indicate to you that there will be no live stream on Thursday of this week due to the United States holiday. And then next week, we're going to talk about some instances of applied statistics in data science. And due to your guys' requests, we're going to start kind of couching a lot of our discussions in the context of applied mathematics, applied statistics, applied linear algebra. And I think that's actually going to be a really cool way to study data science together. So, yeah, you have the finished version of this. If you just refresh the page, you should have the link to the completed version of this notebook. Please reach out to me via email or via Twitter or any other way if you have feedback about how I could make these lessons better. And if I don't hear from you, I will see you back here on Tuesday the 9th. Tuesday the 9th. Okay. Thank you, Haley. Thank you, Caesar. I hope you've liked today's discussion, and I'll see you next week. Thanks, everyone.